This is a review of the endomembrane system through a homework question that you had in your third problem set, number 5, where you were supposed to go and look at the path of a soluble glycoprotein through the endomembrane system through its production, and also to follow the path of a transmembrane or integral protein. A glycoprotein through the same endomembrane system. Now, first, let's review soluble versus transmembrane. Soluble means that it is free floating, it is dissolved in the fluid. A transmembrane means it's a protein that's attached to the membrane. Also, glycoprotein is a protein that has carbohydrate additions, and it receives these through a process called glycosylation. And these carbohydrates are added via enzymes that are found in the compartments of the endomembrane system. Okay, so let's first take a look at the path of a soluble glycoprotein. And the glycoprotein first has to be produced, and a glycoprotein is produced in the rough endoplasmic reticulum, and it is made by ribosomes, which are bound to the endoplasmic reticulum, and that's why it is called rough. So these are ribosomes. They use RNA as the information in order to produce a protein, and when they produce a protein, that protein is now, if it's a soluble protein, it is going to be completely within the lumen of the ER. Now, in the rough endoplasmic reticulum, not only is the protein produced by the ribosomes, but also there are enzymes in there that can perform glycosylation reactions. In other words, these proteins get carbohydrate additions on them. Now, the carbohydrate additions that they get are all the same. It's a core amount of carbohydrates. These are sugars. And so the core additions are placed in the endoplasmic reticulum. So at this point, what we have here is, if I were to label this, is that this right here, okay, that is a soluble glycoprotein. It has just been produced and glycosylated. It is not mature yet, and that is going to be the role of the remainder of this pathway. So once this glycoprotein has been produced, the carbohydrate additions have been placed on it. You also learned any disulfide bridges would occur in the proteins in this compartment. It now gets packaged, and when it gets packaged, it goes into a vesicle. And if it was in the lumen of the ER, it is going to be in the lumen completely within the completely within the lumen of the vesicle. I'm just showing one here as an example. Now this vesicle, this transport vesicle, will then fuse with the membrane of the Golgi. And so what was inside the ER and now was inside the lumen of the transport vesicle is now within the lumen of the Golgi apparatus. So let's go ahead and draw this now inside the lumen of the Golgi apparatus. Now it enters, if you recall, on the cis face of the Golgi, and through cisternal maturation, which we discussed in class, that protein will find itself on the transface, where it will be packaged, sorted, and packaged for export. Now what's happening as it is traveling from the cis phase to the trans phase is the compartment that contains that glycoprotein, the enzymes that add the carbohydrates are going to change. And so what will happen is the carbohydrates will be clipped and altered as it goes from the cis to the trans phase. Oops, again, wrong color. So by the time it reaches the trans phase, the carbohydrate additions will be altered than how it started. So now this is this soluble glycoprotein that is now matured in the Golgi apparatus will be packaged in a vesicle and okay, I couldn't all fit it in there. And when it's packaged in this vesicle, it will be sorted. And in this example, it is being sorted to go to the plasma membrane. So here it is in here. Same idea with its matured carbohydrates on there. And this is now going to be a secretory vesicle, since it is destined for the plasma membrane. Now it will fuse with the plasma membrane, and everything that was in the lumen of the secretory vesicle is now going to be exposed to the extracellular surface, the extracellular side of the cell. So what we're going to get then from this is that this protein will exit the cell, and now will be what we call secretion. It has now been secreted to the extracellular side of the membrane. So once again, we have the protein was produced in the endoplasmic reticulum. It was modified with carbohydrates because of the enzymes that were within the lumen of the rough ER. It was packaged into a transport vesicle which fused to the cis face of the Golgi apparatus. Those carbohydrate additions were modified as the protein matured from the cis phase to the trans phase because of enzymes that were in those compartments. It was packaged into a secretory vesicle, which then fused with the plasma membrane, and the glycoprotein was secreted. Okay, so this is a soluble glycoprotein, but what about a transmembrane glycoprotein? Well, in a transmembrane glycoprotein, the protein is going to still be produced by ribosomes, right, that are on the surface of the endoplasmic reticulum. So it's still in the rough ER. But the difference is that it is going to be a transmembrane protein that will be produced. In other words, it will extend, and part of that protein will be facing the ER lumen, and part of the protein will be facing the cytosol. And it is only the part of the protein that is facing the cytosol that is going to be able to receive the carbohydrate additions, because that's where the enzymes are. So again, I'm going to show the carbohydrate additions on these transmembrane glycoproteins. Okay, so this is a transmembrane, meaning the protein crosses the membrane. It has carbohydrate additions, that's the glyco, and it is a protein. It is immature, so like the protein that was the soluble glycoprotein, it is going to have to go through modifications of its carbohydrates. Now that the protein has been produced, and it has its core glycosylation on it, it will need to be packaged into a transport vesicle. And what was inside the lumen of the ER will be in the lumen of the transport vesicle. And this transport vesicle is going to then fuse with the cis face of the Golgi. When it fuses with the cis face of the Golgi, oops, wrong color. When it fuses with the cis face of the Golgi, the carbohydrates are going to be facing the interior. Now, as this protein matures, it starts out as an immature protein. As it matures, as it is taken from the cis Golgi by cisternal maturation to the trans Golgi, the carbohydrates on the protein will mature. So let's first show it as it enters. And then when it's about in the middle, the enzymes right in the chamber alter so that different carbohydrate additions can be made and modified. And then by the time it is in the trans Golgi, where the protein is going to be sorted and shipped to its final destination, the carbohydrates are considerably different than what they used to be. And so this mature glycoprotein is going to be packaged and sorted into a transport vesicle. And since we've already said that this is bound for the plasma membrane, it's going to be a secretory vesicle. So here we go. And anything that was inside the lumen of the Golgi is now in the lumen of the transport vesicle. It's the only place the carbohydrate additions are on. They're not on the other side. They're not on the cytoplasmic side. So that now when this vesicle fuses with the plasma membrane, the carbohydrate additions are all facing extracellularly. So this transmembrane glycoprotein, which I'll show here, situated in the plasma membrane, will have its carbohydrates facing the extracellular side. So, in review, the protein was produced on ribosomes bound to the ER. There was one or more hydrophobic stretches in the protein, so it got stuck in the ER membrane. The enzymes within the ER lumen added carbohydrates to that protein, and the protein was packaged into a secretory vesicle, into a transport vesicle. The transport vesicle then fused with the cis face of the Golgi. This immature glycoprotein then matures to a mature glycoprotein by the time it reaches the trans face of the Golgi by cisternal maturation. And this mature protein has its mature carbohydrate additions on it. It is packaged into a secretory vesicle, and the secretory vesicle fuses with the plasma membrane such that this transmembrane glycoprotein, which has a portion of it that faces intracellular and a portion of it that faces extracellular, it is only the portion that faces the extracellular side that has the carbohydrate additions. And this is how the cell is able to have transmembrane proteins in its plasma membrane. For example, any of those channels or transporters or the sodium-potassium pump, this is how it all gets there.